I believe that to truly understand something, it is important to go back to how it came into existence, as long ago as 1616, when William Harvey establishes that blood flows through a closed system. Earlier, it was thought that arteries contained air and that blood freely dissipated through tissues as and when they needed it and circulated back in veins to the liver for purification. But Harvey's work concluded that blood traveled only inside vessels from the heart to tissues in arteries and back to the heart in veins. This was extremely important for transfusions because it meant that fluid could not just be injected into any part of the body but rather had to be injected into this closed system. Following this, in 1665, Richard Lower began experimenting with blood transfusions from animals to humans. But 2 years later, Jean-Baptiste Denis, the physician to King Louis XIV, recorded signs and symptoms of hemolysis in his blood transfusion experiments, for which he was jailed, and blood transfusions from animals to humans were banned. Thus, the field of blood transfusion remained stagnant for another 150 years till 1829, when James Blundell, an obstetrician, noted that acute blood loss in women following childbirth could be managed with blood transfusions. But he insisted that it had to be with human blood. We know why this is in the current day and age, but it took another 50 years for people to understand this insistence when Leonard Landoy examined black urine in animals who had received a heterologous blood transfusion and unraveled the fact that blood cells from one animal species can hemolyze when they come in contact with the serum of a different animal species. And that is why animal blood should not be transfused to humans. At this time, it was thought that incompatibility arose due to difference in species, because it took another 25 years for Karl Landsteiner to discover the ABO blood group, and another 40 years for him to discover the Rh blood group along with Alexander Weiner. But between 1920s to the 1960s, a lot of other blood groups were discovered, mostly after searching for an explanation for an unpredicted blood transfusion reaction between bloods that were known to be both ABO and Rh compatible. Around the same time as Rh blood group was discovered, Philip Levine theorized that hemolytic disease of the newborn was due to incompatibility of maternal and fetal blood. He then demonstrated this by reacting the red blood cells of a husband and later their firstborn child's with the serum of the mother and observed that the red blood cells were destroyed. But because this hemolysis was so prompt, the immunoglobulin responsible was probably IgM, and this was an obstacle for early diagnosis because in hemolytic disease of the newborn, a mother forms antibodies to her fetus's red blood cells due to an incompatibility. But these antibodies don't act upon her firstborn child. Rather, they act on her secondborn child. Because of this delay in action, they cannot be IgM in nature. Furthermore, the secondborn child experiences a massive hemolysis as soon as they are born, which means the antibody has to be able to traverse the placenta, which also means it could not be IgM in nature, so this had to be an IgG antibody. And this was another obstacle because most IgG antibodies were non-agglutinating. So it would be challenging to detect their action in vitro diagnoses. Around this time in 1943, a veterinary doctor named Robin Coombs was attempting to devise a diagnostic test for Pfeifferella malae, which is now known as Burkholderia malae, a potentially fatal disease for both horses and humans. His work was critical as this was wartime when bacterial infections were becoming a common cause of death in soldiers. He devised a sensitive serum test that later influenced his infamous Coombs test. He found that horses exposed to the bacteria produced a powerful agglutinating serum protein called conglutinin, but that this protein did not cause agglutination of red blood cells unless it was also attached to hemolyzing complement proteins. So in this test, exposed animal serum containing conglutinins were incubated with lab-generated hemolyzing complement proteins to allow for the interaction. And then later, reagent red blood cells were added to the mixture, which would form clumps and then burst, confirming the diagnosis of Glander's disease. So that is why when Coombs and his colleagues tried to explain the phenomenon of blood incompatibility that had just recently been described by Levine, he drew inspiration from his work as a vet. What was known was that red blood cells do not agglutinate in the body due to a negative charge on their surface that causes repulsion, a phenomenon we now know as the zeta potential. Sialic acid produces this repulsion. Furthermore, IgG antibodies are too small to cover the minimum distance between red blood cells, whereas IgM antibodies are large enough to cover the distance to cause agglutination. This is another reason why it was concluded that Philip Levine's test of maternal serum and paternal blood cell hemolysis had shown IgM antibodies. Also, in the 1940s, it was only recently established that the nature of antibodies was gamma globulin, as it was earlier thought that complement proteins, which are beta globulins, were antibodies. And so Robin Coombs used a mix of some old and new information to theorize that some red blood cells may have antibodies, by which he meant immunoglobulins and complement attached to them that don't normally cause agglutination but have the potential to cause it if they interact with another substance. Thus, they are normally incomplete for not causing a visible reaction. Note that incomplete antibodies are now known to be IgG type, which we have established are too small to cover the gap between two red blood cells and so cannot possibly cause agglutination on their own. But if a substance X could bridge this gap and complete this antibody, there could be a visible reaction. Coombs soon worked out that the substance X was none other than an antibody directed towards this incomplete antibody on the red blood cell surface. This was later refined to become the Coombs test. So what they did was inject immunoglobulins and complement proteins from many different human samples into a rabbit. In fact, they injected them into many different rabbits so that different rabbits could create many different antibodies against the human plasma proteins, which could all be combined together to form the Coombs reagent. This is used daily in blood banking because these rabbit-produced antibodies can complete the incomplete antibodies on the red blood cell surfaces and produce a visible agglutination reaction. To perform the Coombs test, we draw blood from a patient and can either directly test red blood cells for the presence of antibodies bound to them, or we can indirectly test for antibodies circulating in the serum that could potentially bind to the red blood cells. We could add the Coombs reagent to red blood cells and the rabbit antiglobulins will agglutinate cells that had the incomplete antibody. Or we could add the Coombs reagent to the serum and let the mixture incubate along with reagent red blood cells. And the reagent red blood cells would form clumps if the serum had antibodies that could not only interact with the rabbit antiglobulins but also bind to the red blood cells. These are the direct and indirect Coombs tests, respectively. One can imagine how many applications these tests can have. The serum can be tested for the presence of circulating antibodies such as those produced in transfusion reactions, certain leukemias, lupus, and infections like brucellosis, infectious mononucleosis, and syphilis, which interestingly was discovered to be diagnosable in the first paper Coombs published after a newborn who tested positively for incompatibility with maternal blood had died despite receiving interventions and was later found to have syphilis post-mortem. So that infant had falsely tested positive for hemolytic disease of newborn, instead testing positive for congenital syphilis. And the blood cells can be tested for incomplete antibodies attached to them that cause hemolysis, such as in autoimmune and drug-induced hemolytic anemias, hemolytic disease of the newborn, and transfusion reactions. Interestingly, this test shortly led to the discovery of a new blood group by a colleague of Coombs named Rob Race. This blood group was named after the mother who lost a baby to hemolytic disease of the newborn as the Kell blood group. Being highly customizable as well as inexpensive, Coombs imagined that this test would be highly useful for diagnostics in developing countries. And so, this test was revolutionary. But it is not without faults. To begin with, the rabbit antibodies are against IgG antibodies and would not react to any other antibody type such as the IgA type, which is a common cause for some autoimmune hemolytic anemias. Moreover, on average, a minimum of 200 incomplete antibodies must be present on a red blood cell surface to cause significant agglutination. But nevertheless, the Coombs test has laid groundwork for many more diagnostic tests. Lastly, to honor the late Robin Coombs, I must mention that he has never approved of his name being used for this test, because after publishing his work, he discovered that it had already been proven by an Italian scientist named Carlo Moreschi 40 years ago, way back in 1908. And so, I hope you enjoyed this lecture on the antiglobulin test. Thank you.